Welcome to our deep dive into pairwise sequence alignment. In the world of bioinformatics, comparing biological sequences is fundamental. Whether it's DNA, RNA, or protein sequences, understanding their similarities and differences helps us uncover evolutionary relationships, predict functions, and identify mutations. Pairwise sequence alignment is the process of comparing two sequences to find the best matching regions between them. This technique is a cornerstone in genomics and molecular biology, providing the basis for more complex analyses. In this video, we'll break down the basics, explore key algorithms, and look at real-world applications. Let's get started by understanding what pairwise alignment really means. Sequence alignment is a method used to arrange two sequences of DNA, RNA, or proteins to identify regions of similarity. These similarities may be a result of functional, structural, or evolutionary relationships. By aligning sequences, researchers can pinpoint conserved regions, detect mutations, and infer phylogenetic relationships. Pairwise alignment specifically focuses on comparing just two sequences at a time, making it a foundational tool before moving on to more complex multiple sequence alignments. Understanding sequence alignment is essential for anyone working in genomics, as it underpins many analyses in the field. You might wonder, why is sequence alignment so important? Well, aligning sequences allows scientists to identify conserved regions that may be crucial for biological function. It also helps in detecting evolutionary changes such as insertions, deletions, or substitutions. By comparing sequences, we can predict the function of unknown genes, trace the origins of diseases, and even design targeted therapies. In essence, sequence alignment is a powerful tool that bridges the gap between raw genetic data and meaningful biological insights. Pairwise sequence alignment comes in two main types: global and local alignment. Global alignment tries to align the entire length of both sequences, making it ideal when the sequences are similar and of comparable length. Local alignment, on the other hand, focuses on finding the best matching region within the sequences, which is useful when sequences are only partially similar or contain conserved domains. Choosing the right type depends on your research question and the nature of your sequences. Global alignment is designed to align two sequences from end to end. This method is best when the sequences are of similar length and are expected to be closely related. The Needleman-Wunsch algorithm is the classic approach for global alignment, using dynamic programming to find the optimal alignment across the entire length of both sequences. This technique is widely used in comparing genes or proteins that share a common ancestor. Local alignment, in contrast, focuses on finding the best matching region within two sequences. This is especially useful when sequences are only partially similar or contain conserved motifs. The Smith-Waterman algorithm is the go-to method for local alignment, allowing researchers to identify regions of high similarity even within otherwise divergent sequences. Local alignment is crucial for discovering functional domains and motifs within genes or proteins. To determine the best alignment, we need a way to score how well two sequences match. Scoring systems typically reward matches, penalize mismatches, and assign penalties for gaps introduced during alignment. The choice of scoring matrix, such as PAM or BLOSUM for proteins, can greatly influence the results. By carefully selecting scoring parameters, researchers can tailor alignments to their specific needs and biological question. Introducing gaps during alignment allows for insertions or deletions, but too many gaps can distort the results. That's why gap penalties are used to discourage unnecessary gaps. There are two main types: linear and affine gap penalties. Linear penalties assign a fixed cost per gap, while affine penalties distinguish between opening a gap and extending it. Choosing the right gap penalty is crucial for accurate alignment. Substitution matrices are essential tools in sequence alignment, especially for proteins. They provide scores for aligning different amino acids, reflecting how likely one is to substitute for another through evolution. Common matrices include PAM and BLOSUM, each with its own strengths depending on the evolutionary distance between sequences. The right matrix helps ensure biologically meaningful alignments. Needleman-Wunsch algorithm is a dynamic programming approach for global alignment. It systematically explores all possible alignments, filling in a scoring matrix to find the highest scoring path from start to finish. This guarantees an optimal alignment, though it can be computationally intensive for very long sequences. Despite its age, it remains a gold standard for global alignments. For local alignment, the Smith-Waterman algorithm is the method of choice. Like Needleman-Wunsch, it uses dynamic programming, but it allows for the alignment to start and end at any position within the sequences. This flexibility makes it ideal for finding conserved domains or motifs, even when the overall sequences are quite different. Dynamic programming is a key technique in sequence alignment algorithms. By breaking down the problem into smaller subproblems and storing intermediate results, dynamic programming ensures that the optimal alignment is found efficiently. This approach is the backbone of both global and local alignment algorithms, making complex comparisons computationally feasible. Visualizing sequence alignments helps researchers quickly spot similarities, differences, and conserved regions. Tools often display alignments as rows of letters, with matches, mismatches, and gaps clearly marked. Visualization makes it easier to interpret results and communicate findings to others, whether in research papers or presentations. Pairwise sequence alignment is widely used in genomics. It helps identify homologous genes, annotate genomes, and detect genetic variations. By comparing sequences from different organisms, researchers can trace evolutionary relationships and discover genes responsible for specific traits or diseases. The insights gained from alignment drive advances in medicine, agriculture, and evolutionary biology. Aligning protein sequences is crucial for understanding structure and function. By comparing proteins, scientists can predict functional domains, identify active sites, and infer evolutionary relationships. Protein alignments also aid in drug design as they reveal conserved regions that may serve as drug targets. Substitution matrices like BLOSUM are especially important in protein alignment. DNA sequence alignment is fundamental for gene discovery, mutation detection, and evolutionary studies. By aligning DNA sequences, researchers can identify conserved genes, regulatory elements, and genetic variations. This information is vital for understanding genetic diseases, tracing ancestry, and improving crop varieties through selective breeding. RNA sequence alignment is essential for studying gene expression and regulation. By comparing RNA sequences, scientists can identify conserved motifs, alternative splicing events, and non-coding RNAs. These insights are crucial for understanding how genes are regulated and how they contribute to development and disease. One of the main uses of pairwise alignment is homology detection. By aligning sequences, researchers can determine whether two genes or proteins share a common ancestor. Homologous sequences often have similar functions, so detecting homology is key for predicting gene or protein function in newly sequenced organisms. Pairwise alignment is invaluable for mutation analysis. By comparing a reference sequence to a sample, researchers can identify single nucleotide changes, insertions, deletions, and other mutations. This is critical for diagnosing genetic diseases, tracking the evolution of pathogens, and developing personalized medicine strategies. Aligning sequences is the first step in building phylogenetic trees, which depict evolutionary relationships among species. By identifying conserved and variable regions, researchers can infer how species are related and estimate divergence times. Pairwise alignment provides the foundation for these evolutionary studies. Pairwise alignment is at the heart of database searching tools like BLAST. When you input a sequence, these tools align it against millions of database entries to find the best matches. This helps researchers quickly identify unknown sequences, annotate genes, and discover new functions. There are many software tools available for pairwise sequence alignment. Popular options include BLAST, Emboss, and Clustal Omega. Each tool has its strengths from speed to accuracy and is suited to different types of analyses. Choosing the right tool depends on your specific research needs and the size of your datasets. To perform sequence alignment, sequences must be provided in specific formats. Common formats include FASTA and GenBank. These formats ensure that software tools can correctly interpret and process the sequence data. Understanding input formats is essential for successful alignments. After running an alignment, interpreting the output is crucial. Results typically include aligned sequences, scores, and sometimes graphical representations. Understanding what these results mean helps researchers draw meaningful conclusions and plan further experiments. While powerful, pairwise alignment has its limitations. It can be computationally intensive for very long sequences and may not capture complex relationships present in multiple sequences. Additionally, the quality of the alignment depends on the choice of scoring matrices and gap penalties. Being aware of these limitations helps researchers interpret results appropriately. Pairwise alignment is often a stepping stone to multiple sequence alignment, MSA, where more than two sequences are compared simultaneously. MSA provides deeper insights into evolutionary relationships and functional conservation, but it builds on the principles established by pairwise alignment. There's often a trade-off between speed and accuracy in sequence alignment. Fast algorithms may sacrifice some accuracy, while more accurate methods can be slower and require more computational resources. Choosing the right balance depends on your research goals and available resources. To speed up alignment, heuristic methods like BLAST use shortcuts to find high-scoring matches quickly. While not guaranteed to find the absolute best alignment, these methods are fast and effective for searching large databases. Heuristics are essential for handling the massive scale of modern genomic data. Pairwise alignment provides valuable evolutionary insights. By comparing sequences from different species, researchers can identify conserved genes and trace the history of genetic changes. These insights help us understand how life has evolved and adapted over time. Aligning sequences is a key step in functional annotation. By comparing a new sequence to known genes or proteins, researchers can predict its function, identify domains, and suggest possible roles in the cell. Functional annotation accelerates the discovery of new genes and pathways. Comparative genomics relies heavily on pairwise alignment. By comparing genomes from different species, scientists can identify conserved and unique genes, study genome organization, and uncover the genetic basis of traits. This field has revolutionized our understanding of biology and evolution. Pairwise alignment plays a role in personalized medicine. By comparing a patient's genetic sequence to reference genomes, doctors can identify mutations linked to disease and tailor treatments accordingly. This approach is paving the way for more precise and effective healthcare. In drug discovery, aligning protein sequences helps identify conserved drug targets and predict how drugs will interact with proteins. This accelerates the development of new therapies and improves the chances of success in clinical trials. Pairwise alignment is used in agriculture to improve crop varieties and livestock breeds. By comparing genetic sequences, researchers can identify genes linked to desirable traits such as disease resistance or higher yield. This knowledge guides selective breeding and genetic engineering efforts. In forensic science, pairwise alignment helps match DNA samples from crime scenes to suspects. By comparing sequences, forensic experts can establish identity, solve crimes, and exonerate the innocent. Sequence alignment is a powerful tool in the pursuit of justice. Environmental scientists use pairwise alignment to study biodiversity and monitor ecosystems. By comparing DNA from different organisms, they can identify species, track invasive species, and assess ecosystem health. This information is vital for conservation and environmental management. Pairwise alignment is crucial for tracking pathogens during outbreaks. By comparing viral or bacterial genomes, scientists can trace the source of infections, monitor mutations, and inform public health responses. This approach has been especially important during recent global health crises. Pairwise alignment is a valuable teaching tool in biology and bioinformatics courses. By working through alignment exercises, students learn about molecular evolution, genetic variation, and computational biology. These skills are increasingly important in today's data-driven world. Despite its power, sequence alignment faces challenges. Highly divergent sequences can be difficult to align accurately, and repetitive regions may cause ambiguity. Advances in algorithms and scoring systems continue to improve alignment quality, but challenges remain, especially with large and complex genomes. The future of pairwise alignment is bright. New algorithms, faster computers, and better scoring systems are making alignments more accurate and accessible. Integration with machine learning and artificial intelligence promises to unlock even deeper insights from sequence data in the years ahead. Artificial intelligence is beginning to play a role in sequence alignment. Machine learning models can predict optimal alignments, identify patterns, and even suggest new scoring systems. This integration is opening up exciting possibilities for faster and more accurate analyses. Cloud computing is transforming how sequence alignments are performed. By leveraging powerful remote servers, researchers can align massive datasets quickly and efficiently without the need for expensive local hardware. Cloud-based tools are making advanced bioinformatics accessible to more scientists worldwide. Modern alignment tools feature user-friendly interfaces that make it easy for researchers of all backgrounds to perform alignments. Intuitive design, clear instructions, and helpful visualizations lower the barrier to entry and encourage more widespread use of these powerful techniques. The bioinformatics community offers a wealth of resources for learning about pairwise alignment. Online tutorials, forums, and open-source software make it easy to get started and stay up to date with the latest developments. Collaboration and knowledge sharing drive innovation in this rapidly evolving field. Wrap-up pairwise sequence alignment is a foundational technique in bioinformatics, enabling researchers to compare sequences, detect mutations, and uncover evolutionary relationships. From basic research to practical applications in medicine, agriculture, and beyond, alignment tools continue to shape our understanding of biology. As technology advances, the power and accessibility of sequence alignment will only grow, opening new frontiers in science.